Hello and welcome everybody to my channel ImmuniBioEntrance, and today we will be discussing CSIR PYQs of the year 2020. I will be discussing the genetics section. If you are new to my channel, please do like, share and subscribe. Then next one. Coming to the year November 2020, shift 1. What kind of inheritance is indicative in the pedigree chart shown below? So this is the pedigree chart. Now they have given options as Y-linked, X-linked dominant, X-linked recessive, autosomal dominant. Now Y-linked, it is not Y-linked because female does not have Y chromosome, but the pedigree females are deceased. Over here, female is deceased. Now if it was X-linked dominant, then 3-2, that is over this one, this female would have been affected. Okay. Now X-linked recessive, then 2-1 and 2-2 are deceased, so their offsprings should also have the disease, but it is not so. This case. So it is the case of autosomal dominant. Answer is option 4. Then next one. Which one of the following correct statement about the difference between AF plus and HFR cell? So the answer here is option 4, that is in F plus cell, F factor is in the form of an episome, while in the HFR cell, F factor is integrated into the bacterial chromosome. So the answer here is option 4. Then next one. Mutation in a is essential for genetic variation. Which one of the following events can lead to variation amongst the gametes produced by males of Drosophila melanogaster? Crossing over does not occur in Drosophila males. Segregation, imprinting. So in 1 and 2, it does not cause recombination. So they have asked mutation, there is no crossing over, means option 3, that is recombination is also not causing variation. So what is left out? Independent assortment. Answer is option 4. So coming to the next question. Cytoplasmic male sterility in plants is caused by mutation in the mitochondrial genome. CMS can be restored by nuclear gene restorer of fertility, which is a dominant character. If a male sterile pea plant is pollinated by a fertile male pea plant with RF in heterozygous condition. So the fertile plant is the female, RF in heterozygous condition, and this is the male sterile pea plant is the male. The progeny obtained will be, so small a will be the female and capital A small a will be the male. Okay, I made a mistake while telling it in the while explaining. So once you cross it, you will get 50% of the progeny as fertile and 50% as male sterile. So the answer here is option 3. A scientist synthesized 4 new chemicals which had mutagenic potential and named them as C1, C2, C3, C4. He tried to analyze the nature of mutations caused by them and obtained the following results. Which one of the following answers describes the mutagenic potential of the chemicals? So if you see here, 2-aminopurine, C1, 2-aminopurine is causing reversion, nitrous acid is causing reversion, hydroxylamine for some is caused reversion. Now 2-aminopurine, hydroxylamine, nitrous acid, all these cause transition mutations. So C1 is caused by transitions. C2, none of them are being reversed by these chemicals. So it is the case of a transversion. C2 causes transversion or large deletion. Now C3, it is reversed by aminopurine and nitrous acid, so it causes transitions. Then C4 is reversed only by acridine orange. So C4 causes single base insertion or deletions because acridine orange causes single base insertion or deletion. So reversal causes, so it is reversed by acridine orange. So C4 is cause single base insertion or deletion. So the answer here is option 1. Then next one. In Drosophila melanogaster, traits A, B, C results from recessive alleles that are located on one of the autosomes. The resulting F1 females were test crossed and the following progeny, total 1000, were obtained. The following conclusions were made of the above data. So this is the case of a 3-point test cross. Now A plus B plus C plus A, B, C are the parentals. Now double crossovers are those with lesser number. So A, B, C plus and A plus B plus C. That is 15 and 10 and 15 respectively. So order of the genes, if you see here, the C is changing position. So C will come in the middle, flanked by A and B or B and A also. So it can be ACB or BCA. So the order of the genes is ACB, that is correct. Order of genes is BCA, that is also correct. Now distance between A and C. Distance between A and C, if you do the single crossover, if you single crossovers, you can see that the distance between A and C is given by 25 plus 35, that is the single crossovers progeny, and plus 10 plus 15 double crossover progeny divided by total progeny into 100, you will get 8.5 centimorgan. Similarly, in the case of B and C, you will get 21 centimorgan. So the answer here, B and C are correct. So answer here is option 3. Then next one. The table given below shows the load score value of 3 different pairs of genes studied for assessing if they are linked pairs. So load score, gene pair 1, load score is 1, gene pair 2 is 2, gene pair 3 is 3. The following conclusions are made on the above data. For gene pair 1, the probability of the genes being linked is 10 times more likely than them assorting independently. Now what have they asked? Which one of the following options or statements that is or are correct? So A is correct. Is 10 times more likely than them assorting independently because load score is log of the odds. Then next one, for gene pair 2, the load score 2 indicates that the probability of the genes being linked is twice more likely, not twice, it is 100 times. So B is not correct. Now C. The gene pair of pair 1 and 2 can both be considered as linked while the genes pair 3 exhibit independent assortment. 3 also shows linkage only, it is load score value is positive. Positive load score value means it shows linkage. Now D. The genes of pair 3 can be considered as linked. That is also correct. Now which one of the following option represents statement that is or are correct is option 3, A and D. Now next one. In mammals, coat color is governed by genes A, B. The coat color is either black or brown depending on whether the genotype is capital B or capital B small b. It is not known which of these genotypes lead to black color and brown colors. The genotype small b results in albino coat color, while further the genotype small c suppresses the expression of coat color, resulting in albino coat color. An albino male was crossed with a brown female, and the resulting progeny had individuals with either black or brown coats. From this observation, it can be inferred that the genotype of the male and the female that are crossed is. So small b and small c result in albino coat color. So if you see 1, capital B, small c into capital B, small b, capital C, you will get black or brown coat upon crossing. So 1 is the correct answer. Answer is option 1. So coming to the year November 2020, shift 2. Now they have given a pedigree here, and they have asked about the trait shown in the above pedigree. So if you see the pedigree, you can see that the female is also affected. So it is not any Y-linked trait as given in option C. Now there is skipping of generation over here, so it is not a dominant trait also. If it was X-linked recessive, then the female got the disease but the male is not having the disease here. So it is not X-linked recessive also. So what is left out? It is autosomal recessive trait. So the answer here is option 2. The next one. A plant that produces disc-shaped fruit is crossed with another plant that produces long fruit. So disc-shaped fruit is crossed with long fruit. All the F1 plants have disc-shaped fruits. So disc-shaped is the dominant character. When the F1 were intercrossed, F2 progeny were produced in the following ratio. 9 by 16 plants in the disc-shaped fruit, 6 by 16 plants spherical fruits, 1 by 16 plants having long fruits. Which one of the following option give correct genotype of the spherical fruits obtained in F2? So this is a variation of the 9:3:3:1 ratio. So that means 2 genes are involved. So capital capital B will be disc shaped, and small b will be long shaped. Now if it was in the 9:3:3:1 ratio, then these are the genotypes that you will be getting. But here it is 9:6:1 ratio. Now 6 you got by addition of these 2 genotypes. That is 3 plus 3 will give you 6. So which of the following option gives the correct genotype of the spherical fruits obtained in F2 plants will be A, B, small b, small a, capital B. So the answer here is option 3. Then next one. The maximum frequency of recombination that can occur between 2 loci, that is 50%. Answer is option 2. It's a very direct question. Then the gene was located on 10p11. This which means the gene was located on P means it is on the short arm of chromosome 10. So short arm of chromosome 10 at G sub band 1 of 1. That is the answer. Answer is option 1. Coming to the next question. The figure given below represents a profile of DNA markers in 2 parents P1 and P2. Progeny F1 from the cross between P1 and P2 and that of gametes produced from F1. So these are P1 and P2, A, B, C, D, E and F, and this is F1. F1 is also having, okay, A is there, E is there, B is there, F is there, C is there, D is there. So F1 is having all the bands from both the parents. These are the DH1 to DH8 are the gametes produced from F1. 8 different patterns DH1, DH8 were observed in the case of gametes. The numbers below DH1 to DH8 indicate the number of individuals observed in each case. Based on the above observation, the following statements were made. Which one of the following have a combination of all correct statements? So first statement, marker B and F. B and F are likely to be allelic in nature. So if in F1 if you see B and F are present, so it is allelic in nature. B1 in F1 if you see there is B, there is F, so it is allelic in nature. Then markers C and D are linked in trans with a map distance of 24 centimorgan. So C and D are present in single parent, and C and D are linked. Okay, we will see that. C and D, here you can see C and D. In DH1 also C and D, DH2 both are not present. DH3 C and D, DH4 C and D, like that. So if you see C and D, they are linked. They are linked but in cis orientation, not in trans. They are found in the same in a single parent. So they are linked and in cis orientation. So option B, statement B is not correct. Now next statement. Marker B assorts independently from marker C. Marker B and C. B if you see over here, B, C, okay. If both are not there in DH2, B, C are present in DH3. Then B and C are present in DH7 also. So they both are linked. So they are not assorting independently. So answer here is only option A is correct. Answer is A only. Then next one. Curled wing, ebony color and sepia eyes are three recessive mutations that occur in fruit flies. The loci for these mutations have been mapped and they are separated by the following hypothetical map distance. The interference between these genes is 0.4. The mutant CUESC fly was crossed with homozygous wild type fly. The resulting F1 females were test crossed and produced 1800 progeny. What number of flies in each phenotype class is likely to be obtained in the progeny of the test cross? So this is a 3-point test cross and they have given the value of interference. Interference is 0.4 and we know interference is 1 minus coefficient of coincidence. That is 1 minus observed double crossovers by expected double crossovers. So expected double crossovers is again given by the formula single crossover between A and B divided by 100 and single crossover between B and C divided by 100 into total progeny. So single crossover between A and B. If you take CU as A and E as B, the single crossover between A and B is 20. So 20 divided by 100 into single crossover between B and C. This is B and SC is C. So that is 12 centimorgan. So 12 by 100 or I can say 12% recombination frequency into 1800. That will give you 43.2. That is expected double crossover. Now coefficient of coincidence we know is equal to 1 minus interference. From this formula, if you rearrange it, you will get coefficient of coincidence as 1 minus interference. So 1 minus 0.4 is 0.6. Now observed double crossovers is equal to expected double crossover into coefficient of coincidence. So that is 43.2 into 0.6, that is 25.92 or approximately 26. So that is from this data we cannot make out the number of progeny that you will get from single crossovers. Okay. So double crossover 26 and non-recombinance will be 1250. If you add up all these, that is in option A, if you add up 1250 plus 334 plus 190 plus double crossover 26, you will get 1800 progeny. So your answer will be option 1. Then next one. In some sheets, horns are produced by an autosomal allele capital H that is dominant in males and recessive in females. H plus individuals are hornless. A horned female is crossed with a hornless male. A horned female is crossed with a hornless male. So this is the horned female H and is crossed with a hornless male. H plus is hornless male. One of the resulting F1 female is crossed with a hornless male. So this is the F1 progeny that you get upon cross between horned female and hornless male, that is H plus. Now you are crossing the resulting F1 female. So this is the F1 female that is crossed with a hornless male. Again crossed with H plus male. What proportion of the male and female progeny of F1 will have horns? So these are the various progeny that you get. Now half of them will be male, half of them will be female, because this is an autosomal recessive trait. So we can take it as half of them will be male, half of them will be female. So male, this H plus is horned, and H plus that is female is hornless. H plus female is hornless, and H plus male is hornless. So from this data you can know that 50% of the male progeny but none of the female progeny will be horned. So 50% of the male progeny, that is, this is male and this is male. So 50% of the male progeny are horned, and none of the female progeny are horned. So the answer here is option B. The next one. E. coli cells were simultaneously infected with two strains of phage lambda. One strain of lambda had a mutant host range that is temperature sensitive and is known to produce clear plaques, genotype HSTC. Another strain of lambda carried the wild type allele genotype H plus, ST plus, C plus. Progeny phages were collected from the lysed cells and were plated on bacteria. The following number of different progeny were obtained. So these are the results of the 3-point test cross. These are the number of plaques. Now what will be the order of the three genes and the map distance between them? So first we will see for the order of the three genes. For that you should know the double crossovers. So 350, 300, they are maximum in number, so they are the parentals, and the least numbered ones, that is 4 and 6, they are your double crossovers. That is H plus, C plus, ST plus is one parent, and HC, ST is another parent. Then H plus, C plus, ST and HC, ST plus is the double crossovers. Now if you compare the double crossovers with the parent, you will see that ST gene is changing position. So it has to come in the middle. So the gene order will be H, ST, C. Now if you see the options, you can see that H, ST, C is present in C only. And they have given the distance also. So if you found out the gene order, and you need not have to calculate for distance between them, because that is the only option that is having H, ST, C as the gene order. But I have done the calculation for distance between H and ST, that is single crossovers plus double crossover into 100 divided by total progeny. So that will be 21 centimorgan. Now distance between ST and C, that will be a number of single crossovers plus number of double crossovers into 100 divided by 1000, that will be 15 centimorgan. So the answer here is option C. Now next one. Body weight of rabbits is determined by pair of alleles at two loci A and B that are additive and equal in their effects. Rabbits with genotype A minus, B minus, B minus, B minus have average 1 kg body weight, whereas individuals with genotype A plus, B plus, B plus have animals with average 3.4 kg in weight. A male rabbit with A minus, B minus, B minus is crossed with a female of genotype A plus, B plus, B plus. What will be predicted average weight of F1 progeny of this cross? So if you see here, this is the cross. They have asked what will be a male. This is the male rabbit and this is the female of genotype A plus A plus B plus B plus. What will be the predicted average weight of F1 progeny of the cross? So this is getting crossed and you are getting the F1 progeny as A plus A minus B plus B minus. Now what is the value of weight of A plus? A plus, if you see, A plus B plus have animals of average 3.4 kg. So weight of A plus here will be 0.85. 3.4 divided by 4, that will be 0.85. Similarly, if you see over here, A minus B minus have average weight of 1 kg. So the weight of A minus alone will be 1 by 4, that is 0.25. Now substitute the values here. So A plus will be 0.85, A minus will be 0.25, B plus will be 0.85, B minus will be 0.25. Add up all of them, you will get 2.2 kg. So the answer here is option A.